Okay, so today's topic is human history and diversity. And the questions we're going to look at are, what does genomics tell us about human diversity, and what does genomics tell us about human history? Now, when I write them this way in this sort of dry scientific language, they might not seem particularly striking, but I just want to remind you that these are some of the most profound questions we can ask about ourselves. That is, who are we and where do we come from? Okay? And so, as you'll see today, genomics is a way of answering those questions in a fairly objective way. And as you'll see, that's sort of an extreme necessity when we ask questions of this type, because the history of people asking and trying to answer questions of this type is they get it wrong in very sort of ethnocentric and racist ways. And so, at least in this case, science performed correctly has the hope of giving us objective answers to some of the questions we care most about. That is, who we are, what is our diversity as a species, and where do we come from? So what I want to start with today is basically sort of a quick tour of wrong ideas about where we come from and how we're divided into groups. And as you'll see on this and the next few slides, I'm putting both race and science in quotes. I'm putting race in quotes because, as you'll see, it's not a concept that should be applied to humans if you look at it objectively. And I'm putting science in quotes because even though the people presenting the ideas I'm about to show you were doing so in what they claim to be in a scientific way, the ideas and the sort of way of generating those ideas are anything but scientific. And that's what I'm going to illustrate to you. So what we're starting with here is a map centered on Europe that was generated by this guy, Joseph Deneker, in 1899. And so he proposed a classification of Europeans into 6 races. And there was some debate at the time about, how many races there are. Are there 3 races? Are there 6 races? Are there more races? And so his classification was of 6 races with 4 sub-races. And you can see the different hatch marks are where each race was supposed to be, and the labels here are what those races are. So you have the Nordic race sort of up here, and so on. Now, what was interesting about Deneker is that although he generated this classification, he himself did not favor a view that sort of gave preeminence to one race over the others. That is, he wasn't trying to rank races hierarchically. And in fact, he didn't like the term race and proposed using ethnic groups as an alternative. But his role in this story was kind of sealed by his decision to declare that there was a Nordic race in Northern Europe, because that declaration, that concept was grasped upon by racists that came after him. And in particular, one of the foremost people who sort of promulgated the view that Northern Europeans were superior than others was a guy named Madison Grant, who basically claimed that the Nordic race was superior and that it was the engine of civilization. And so he had a map that looked like this. So this is Madison Grant. He was an American lawyer. And this is his depiction of what the distribution of races in Europe was. And you can see that here we have Nordic, here we have Alpines, okay, so more southern European areas. There's one thing we'll talk about a little bit later here on the map also is what we call Cro-Magnon, or should probably pronounce Cro-Magnon, which is where very early human ancestors were discovered in France, right there. And you can see, even just sort of in the iconography of how he draws this map, how he viewed the Nordic race, that it was basically starting here, but sort of infiltrating the rest of Europe. It's almost like tentacles expanding into the rest of Europe. And he viewed this as a positive thing, right? That the influence of the Nordic race was sort of making its way through Europe, and eventually people would realize that they were superior and join on, and things would be great. And to give you sort of an example of how he expressed these ideas, I mean, we can look at his ideas plainly now as nothing you would describe as anything but racism. So these are direct quotes from Grant. This is what he says about the Nordic race. The Nordics are all over the world a race of soldiers, sailors, adventurers, and explorers, but above all of rulers, organizers, and aristocrats, in sharp contrast to the essentially peasant character of the Alpines, so the more southern and eastern Europeans. Okay, so there you have it. That's basically the standard sort of northern European supremacist line. What's a little bit more interesting is what he says about the Mediterraneans. So he says the Mediterraneans gave the world the great civilizations of Egypt, Crete, Phoenicia, including Carthage. It gave us, when mixed and invigorated with Nordic elements, the most splendid of all civilizations, that of ancient Greece, and the most enduring of political organizations, the Roman state. So this is kind of an interesting statement that says, well, the Mediterraneans were doing okay, but they really got good when they mixed with the Nordics. So there you have it. That's sort of the origins of sort of Northern European white supremacism. And indeed, in the 1800s, there was sort of a cottage industry of people looking in biology for evidence of racial superiority or inferiority. And one of the sort of incarnations of this was what we now call craniometry, or the measurement of skulls. Okay, and so this is from a book from 1857 called The Indigenous Races of the Earth, written by Knott and Gliddon. Knott was a famous surgeon of the time, actually. He was known for his work on yellow fever. And you can see how, I think you can get the idea of how he's depicting sort of a European, someone of African descent, and a chimpanzee. And I don't think it takes much text. You know, you wouldn't have to read the book to get the general idea of what he was trying to get across here. Okay, and what he was trying to get across, of course, is that here you have the superior, inferior, and you can tell it's inferior because it's so similar to a chimpanzee. And so, again, this is sort of a classic example of pseudoscientific racism. You can see even in sort of how the depictions are chosen that it's not at all a scientific argument being made. You have a picture of a statue here, like not an actual person, an ideal, right? That's supposed to be the European here, the Greek, right? And for what he called the Negro, you have an exaggerated actual person, more actual looking, a different angle, right? So the Greek is facing exactly horizontally, and this picture and this picture are both at an angle, and so that sort of emphasizes their similarity. If you turn this one this way, it would look more similar too. And so these guys weren't the only ones doing this. This was sort of a common way of viewing differences between humans. This is a book from 1839 from Morton. So now Morton, and this just I want to sort of emphasize again that this is sort of mainstream in a way at the time. So Morton was a physician. He was a professor at the University of Pennsylvania. Okay, so if you have a friend at UPenn, you know, call him up and say, you know, way to go. We have more, so you have this guy. Okay, so here's how Morton depicted differences between races. Okay, and so again, this is focusing on the shape of the skull. Okay, and here's the Caucasian in the middle, right? And what's being emphasized is sort of the relative roundness or oblongness, the protrusion of the jaw, and so on. Okay, and here's what he says about these skull differences. He says the African skull is sort of narrow in front and having a protruded jaw, whereas the Caucasian skull is symmetrical and full development. And the Asian skull has a retracted face and exposed cheeks. Now, of course, you could, you know, if you decided that you wanted to declare this the superior and this the inferior, you could say this one has, you know, a bulbous head, right, and stubby jaw, right? So the choice of words is really important here, that he sees this as somehow perfect and this as imperfect, but there's no reason to do that. You could sort of turn it around however you wanted to. And what really betrays what the source of these differences is, at least in Morton's mind, is how he describes the characters that are associated with these skull traits. Okay, so he declares people of African ancestry to be fond of amusements and to yield to their destiny. So if you think about the time at which these statements are being made, you can see this as a sort of pseudoscientific justification for slavery. Right? Whereas Caucasians, of course, have the highest intellectual endowments, are the fairest inhabitants of the planet, and the Asians are ingenious, imitative, and compared to the monkey. Okay, so it's clear what he was trying to do here. And as I said, these were actually quite mainstream ideas, so it wasn't for a long time that views like this were refuted, despite how obvious it looks to us today what was going on here, okay, that these were sort of non-scientific beliefs that were given a scientific sheen to make them sort of seem more legitimate. One of the more sort of egregious, sort of offensive indications of how this idea of racial superiority and inferiority sort of infiltrated the culture was that at around the turn of the century, well, going back, into the 1800s and up until the early 1900s, you had what are now called classified as human zoos. Okay, the idea that people would be on display for other people to go pay and see. Okay, so this was a famous example of a poster advertising a human zoo in London in the 1800s. This was a so-called cultural exhibition in Paris in 1907. Okay, where basically you had people dressed up in what was supposed to be traditional garb in traditional settings being viewed by sort of French upper classes who paid money to see it. This is Stuttgart, Germany, 1928. Okay, human zoo. Concept being that people would be on view for other people to view. Now, in case you think this might have been sort of a European phenomenon and a more progressive democratic place would not have such things, this is a man named Oda Benga who was on display at the Bronx Zoo in 1906. Okay, same exact thing, posing with the chimpanzee for people who went to the Bronx Zoo to see. Okay, so again, this is a weird and offensive mixing of sort of the idea of race and the idea of science, that somehow these are true. Because if you think about it, what does it mean to be on display in a zoo? It means that there's some kind of true biological distinction being made, and one we now know to be false. To give you an indication of how even sort of really, really well-regarded thinkers of their day could be incredibly wrong about race, the best example is Carolus Linnaeus. You might have learned about Linnaeus because he's the guy who invented the way we name species. So when we say Homo sapiens for humans, that goes back to him. This sort of binomial classification where you have a genus name and a species name. Homo sapiens, Drosophila melanogaster, any of the other species, that we've talked about in the class. That goes back to him. He was considered, a brilliant, brilliant biologist. And just to, get an indication of how brilliant he was viewed by his, by other brilliant people, these are quotes from two great philosophers, right, Rousseau and Goethe. Rousseau says, "I know no greater man on earth." Okay, and that's high praise coming from, Rousseau, who is still to this day, a very, very, very highly regarded philosopher. Goethe, with the exception of Shakespeare and Spinoza, I know of no one among the no longer living who has influenced me more strongly. So that's really, really high praise for someone. And a lot of what we learn about Linnaeus today deserves that praise, okay, in terms of his understanding of the diversity of biological organisms and the need for classifying them. But he got humans dreadfully, dreadfully wrong, okay? And so in his book, Systema Naturae, Linnaeus classified humans into 4 groups: Americanus, Asiaticus, Africanus, and Europeanus. And you can see, again, the descriptions he gives to these groups. So This would be Native Americans, okay? Reddish, stubborn, angered easily. Asiaticus, sallow, avaricious, easily distracted. Africanus, black, relaxed, negligent. Europeanus, white, gentle, inventive. Okay, so very similar to, you know, the ideas that Morton was going with. And it's easy for us to sort of almost laugh at these now, but we really have to come to terms with the sort of history and even recent history of really ridiculous, offensive, and dangerous ideas on race that were promulgated in the name of science. For Linnaeus, it got even weirder, okay, because he classified another group of humans called Homo, instead of Homo sapiens or Homo asiaticus, you have Homo ferus, or wild, which included so-called wolf boys. So at the time, there were these famous cases of children who had supposedly been raised not by people, but by wolves, or, you know, out in the wild somewhere. And so there was this famous example of Peter the wild boy of Hanover and the wild girl of Champagne, right? And so Linnaeus, for some reason, decided that this was an entire classification by itself, and that there were, you know, that Peter and the wild girl should be a different, basically, species or type of humans. He also had another group called Homo anthropomorpha, which included like completely made-up creatures. So satyrs, other mythological creatures, and Linnaeus claimed they actually existed. They were in the mythology because someone sometime had spotted them, and they were really ape-like creatures, sort of like what's Yeti or something. And so that was another group of humans. So wrong, dangerous, weird. Okay, so I've already mentioned that sort of it was clear that ideas like this were used as justification for slavery, justification for continued racism after slavery ended. Ideas like this clearly went into sort of Nazi thinking, especially the ideas of Morton about the superiority of the Nordic race. Okay, so this is a poster of the Nuremberg laws, which was basically how in Nazi Germany it was decided whether you were Jewish or German or somewhere in between. And you can see the sort of scientific sheen that's given here. You have pedigrees, basically, of families. And if you had 4 German grandparents, okay, you were considered German, right? If you had either 3 or 4 Jewish grandparents, you were considered Jewish. And if you had 1 or 2 Jewish grandparents, you were considered somewhere in between, with different grades of being a little bit better and a little bit worse. And so again, there's no real scientific basis for saying that someone with 3 grandparents who were Jewish is Jewish. That's not at all a scientific concept. But the display of it in this way, the justifications that went along with this, were couched in the language of science. So this, of course, all raises the question of what's the truth. You know, what is the reality? How different are humans from each other? Can you draw lines around particular groups and say this is a race, they're different from each other? Where do, you know, can actual data be brought to bear on this question? And so just to give you a sense of how the modern view of this has changed is it's clearly now seen as a scientific question. You can see here, for example, that on the American Museum of Natural History's website, where they talk about their Hall of Human Origins, which is a great place to visit if you haven't done so, that you have this double helix here. So this is sort of hinting at what they mean to portray is that science, DNA science in particular, genomics, can actually tell us something about where humans come from and how they're different from one another. So that's what I want to talk about now. Where the data come from, what the data are, what they tell us about ourselves. So to do that, we have to introduce one term, which has a long name and a short abbreviation. So the name is single nucleotide polymorphisms, or SNPs, SNPs. We say SNPs. And a single nucleotide polymorphism is basically a position in the genome where individuals of the same species differ in what DNA base they have. So this is an example from the book where you have a stretch of DNA. This is an actual piece of chromosome, right, in a human being. And one person in the population might have a C, and another person in the population might have a G, and then another person might have a C or a G or whatever, okay? And so if there's a place like this where two people in the human population differ at what letter they have, that's called a single nucleotide polymorphism, or a SNP, okay? And on average, two human beings who are unrelated to one another differ at 1 SNP per 1,000 bases. So again, these are actual pieces of real human genome sequences, okay? And if you actually went along, you would see that they're identical except for this one position here, where there's a C in this individual and a G in this individual, and this is 1,000 bases, that's 1,000 bases. And on average, that's sort of the difference that you see if I took, say, my sequence and someone else's sequence in this room, okay? Now, if I took my sequence and my identical twin brother's sequence, they would be exactly the same, right? And people with different levels of relatedness might have fewer differences than this. So for example, let's do some calculations. So if two random humans differ at 1 SNP per 1,000 bases, and the human genome is 3 billion bases long, on average, how many bases do two people differ at? What's the calculation you want to do? So 3 billion divided by 1,000, right? So the whole genome's 3 billion long. Every 1,000 bases, there's a difference on average. So 3 billion divided by 1,000 is 3 million. Okay, so on average, two people will differ at 3 million places in their DNA, single letter changes, right? So let's take another example. So let's say you have two parents who are unrelated and they have a child, okay? If you take the mother and the child and compare them, how many bases will they be different at? 1.5 million, right? Because the child is going to get half their DNA from the mother, half their DNA from the father. And so the half that's from the mother is identical to the mother, and the half that's from the father is different from the mother. And so you get 3 million divided by 2, or 1.5 million. And you can go on and figure it out for like your first cousin once removed and so on. And pretty quickly, actually, you know, you get past like second cousins, and you get basically the same number, about 3 million differences between two randomly chosen people. Okay? And so these differences are what we can use to actually figure out if there are patterns, if people from a particular area, people with a particular appearance, have more similar DNA than others. And so there's a big, and we can also, it should be said, use data like this to try and figure out the causes of disease and so on, right? Other kinds of things we've talked about, where a change like this in the genome might be associated with a disease, right? If you think about it, though, most of the 3 million differences have nothing to do with disease, right? They actually probably have nothing to do with the activity of any of the genes, because as you saw in a much earlier lecture in this class, the human genome is filled with a lot of junk. Okay, a very small proportion of the genome is actually genes. Okay, and so most of these changes don't take place in genes. They probably don't affect the organism in any observable way, but some fraction of them do. So one, is this working? So one reason to try and catalog SNPs is to figure out human history, and then another reason is to figure out potential causes of human diseases and so on. And they're not completely separate projects, okay? As you'll see, those concepts are actually tied together. So there is a big project called the International HapMap Project, which I think Joshua talked about in his lecture, where a large number of individuals from a few populations were each tested for a large number of SNPs, okay? And so those 4 populations are Europeans from Utah, Chinese from Beijing, Japanese from Tokyo, and Yorubas from Nigeria, okay? And so each individual was tested for 1 million SNPs. That is, it was known already that humans, there are certain places in the genome where humans are different, and then each of these people was tested for which allele they have at those locations in the genome, whether they have the C or the G or the A or the T, so on. Okay? And then these data are really useful for figuring out how genetic differences are sort of distributed within the human population. They're also useful for understanding recent human evolution. Okay? And so if you think about it, when we were talking about crop domestication, I said, well, we can tell when a certain genetic difference was responsible for sort of selection by our ancestors. You know, that maize, you know, became less branched as a plant because this gene changed. The way we did that was by seeing that around that gene, around that location, there was less variation, right? And so you can do the same thing in humans. You can say, well, let's look across the whole genome, and are there places where there's less variation? Because if there are places where there's less variation, those genes were probably selected in our human ancestors because they conferred some kind of advantage. And so you can actually do that, and that's been done. It's not an easy thing to do. It doesn't give really clear answers, but it does give some sort of suggestive hints about what our selective history as a species was like. That is, what traits arose that became advantageous. And so there are about 700 human genes that show evidence of having been selected in the recent past. That is, that have a deficit of genetic variation around them. And by recent past, I mean, you know, about the time scale we were talking about with crop domestication, so 5,000 to 15,000 years ago. And as I said, the way that this inference was made was the same way you do it with domesticated crops, finding places in the genome where there's less variation. Now, what's interesting about humans is the types of genes that show this evidence of selection, okay? And as you know, we can get the types of genes just by looking at the gene sequences, okay? Because nowadays, enough genes have been studied where if we say, this is what the sequence of the gene looks like, we can say, oh, that gene is probably involved in whatever. So it's probably involved in skin color, or hair texture, or bone structure, or digestion, taste, smell, detoxification, and so on. So these are the categories of genes that tend to have this signature, this evidence of being recently selected, okay? And so these ones here are all really interesting. So digestion, taste, smell, detoxification, because these are things you can sort of think about in terms of what was also going on around this time, which is that our ancestors began living in civilizations and cultivating plants, okay? So you can think about the massive change in diet that must have occurred when people started growing crops, raising animals, eating dairy, and so on. And those put pressures on certain genes to change, basically, so that advantageous versions of those genes arose that swept through the population of humans because they conferred some kind of advantage. So, for example, the ability to detoxify compounds that might have been found in some of the plants that became a bigger and bigger component of the diet, or the ability to digest certain things. So I gave you the example of lactose tolerance, okay? So that's a mutation that shows evidence of selection because when some of our ancestors started eating dairy, it became advantageous to actually be able to digest lactose as adults, okay? And then brain development is also an intriguing one to think about, right? That somehow in the course of becoming human, our species had changes in our genes that basically made us who we are, made us smarter, at least in some cases. So sperm production is an interesting one. So this is a pattern you see in all animals, basically. If I give you an animal, I say, go study it and find me the genes that are under selection, you would find sperm production genes. And it's not really clear why this is. One of the ideas is that the act of mating actually presents an immunological challenge for a lot of organisms, that it's a chance to sort of transmit diseases, basically. And so it's thought that maybe there's this arms race going on between diseases and the ability of sperm to sort of not succumb to those diseases or be weakened by them. And so you have this constant arms race going on where the sperm has to constantly change so that it's not attacked by some disease before it can fertilize an egg. But that's just one hypothesis. It's not really known why that category always shows up. Okay. And I imagine that as the amount of data that's collected on human genetic differences increases, this kind of study is just going to become more and more common, and the sort of resolution of our understanding of the evolution of these traits is going to become even greater, that this will be sort of an expanding line of research in the near future. But I want to get back to the question about race. Okay, so what is the human genome and the amount of variation and the distribution of variation of the human genome say about race as a concept? So these are two guys who don't often agree, right? These are the two guys who are racing to sequence the human genome by different methods. Okay, but one thing they do agree on is that when you look at the human genome and the distribution of genetic variation, it gives you no support for the idea that race is a valid scientific concept. Okay, so what Francis Collins says is the concept of race has no genetic or scientific basis. What Craig Venter says, race does not exist at an individual level in science or in the genetic code. Okay, and so what are the data that they can actually use to make these statements? So one of the studies that was done to get at this very question, is race a valid concept, was similar to the HAP map, but instead of taking 4 populations, just 4 populations with a lot of individuals, and looking at millions of SNPs, they invested their resources differently. They took many more populations with fewer individuals and fewer SNPs, but the idea was the same. They wanted to catalog where the genetic variation was in different parts of the world. Okay, so what's shown on this map, everywhere there's a dot, is where a population was sampled. So there are 52 dots on this. There are 52 populations that were sampled, 261 individuals total. And for each one of these, they sampled genetic variation not as extensively as HapMap, not at 1 million SNPs, but at fewer, but still enough to get the pattern of genetic variation across the globe. Okay? And the way this study represented that pattern of variation is a little bit complicated, but I think it makes actually a nice way of viewing the data. Okay, and so I want to go through this a little bit slowly. So this is their graph of what the genetic variation looks like across the globe. Okay, and so you actually have the globe, right? And so this is a 3-dimensional plot. So one axis is longitude, and another axis is latitude. So you can think of just sort of a map, right? But instead of the map being flat, it sort of has this curvature to it, this height to it, because what they're doing actually is at every place where one of these populations was sampled, they're saying how similar is the genetic variation you see there to what you see in Asia. Okay, so they have one reference population in Asia, and so you can see here's Asia right here, and so that's very high on the graph. Okay, a lot of shared ancestry with Asia. Because these are Asian populations. And the farther away you get from Asia, you have less shared ancestry. Okay? So that's part of the message, that if you look at genetic variation across the globe, you do see differences. Okay? Different populations have different alleles that are common or rare in them. Okay? Asian populations tend to have certain alleles that are more common. African populations tend to have certain alleles that are more common. Australian populations tend to have alleles that are more common and less common, and so on. Okay? So that's part of the message, that there is information on geographic origin in genetic variation. And we'll talk more about this as we go on in this lecture. But the more important point is that there are not discontinuities. Okay? So one of the things that laying out the data like this shows is that it's a gradual difference across the globe. Okay? So another way of thinking about it is that there's no place where I can say here that Asia ends. Okay? I can't draw a line here and say, aha, all of these populations are so similar to each other to the exclusion of these populations that I'd want to draw a circle around these and say that's the Asian race. Okay? Because basically, the farther you get away from any particular point, geographic location, the more the genetic variation drops. But it does so in a very gradual way. Okay? Similarly, you can't just draw a line around Africa and say, this is the genetic variation that's common in Africa to the exclusion of everywhere else. And actually, as you'll see later in the lecture, to a large extent, the genetic variation across the world is basically a subsample of what's in Africa. Okay? So that's a really important concept, that there's this gradual change in genetic variation across the globe, not discontinuities where you can say, here's Asians as a race, here's Africans as a race, here's Europeans as a race. You can't do that. Okay? So just to summarize, because I think these are some really important points where science, just looking at DNA can actually give us an answer that's useful. Okay? Most genetic differences among humans are found within populations rather than between populations. So what does this mean? This means that on this graph, basically what you're looking at is sort of how rare or common particular alleles are. But in almost every population, you have individuals with almost every allele. That is that genetic variations exist all over the place. And what you see different across the globe is not having certain alleles and not having certain alleles. It's tending to have certain alleles and tending not to have certain alleles. That it's a probability thing. There are many people living in this part of the world that share genetic variation with people living in this part of the world, okay? But those might be alleles that are common here and rare here. Another way of saying this is how one of my graduate school advisors used to say it, is imagine a worst case scenario where a nuclear war is instigated, okay, and nobody survives except the population of Papua New Guinea, okay? So, somewhere around here is all we have left, okay? If that terrible thing happened, over 80% of the world's genetic diversity would still exist, okay? And the reason is that even though certain alleles are common in Papua New Guinea and certain alleles are rare in Papua New Guinea, they're all there, okay? There are very few alleles that exist in one part of the world that don't exist somewhere else. That's what we mean by saying that most of the genetic differences among humans are actually within each population. Each population has a large representation of all of the genetic differences that exist across the whole world. But allele frequencies, so the tendency for certain alleles to be common or rare, do differ across the globe. So there is indeed information in our genes about our ancestry, and we're going to talk about this as we go on in the lecture. The big conclusion is that traits historically used to define human races, for example, skin color, facial features, and so on, are not typical of the genome. Okay? So the things that we recognize because we as a species are good at trying to distinguish, right? Trying to make patterns where they might not even exist. Those things, those visual traits, are not typical of the genome. Okay? And what that means is that very few genes actually are good indicators of racial identity. Racial in the sense of sort of an invalidated scientific concept. And so I didn't assign this article, but I recommend it. It's just a one-page article that talks about these ideas and what sort of modern genomic view of our diversity tells us about race as a concept. Okay. So what I want to switch to now, actually, is this idea that ancestry still is evident in our genes and what uses that has for us and what that can tell us about the history of our species. Okay. So these are images from an article that's in your required reading. And these are different people who have had their sort of genetic ancestry figured out some way using DNA. Okay? And so you can see you get basically what are these quantitative estimates of what your ancestry is. So this individual who you hear a little bit more about in a second, when they got their test done, and we'll talk a little bit about what the tests are like, they found out they had 80% European ancestry, about 10% North African ancestry, and about 10% Native American ancestry. Okay? This individual had about 60% West African, 40% European, 3% Native American, and this individual had about 83% West African, 10% British Isles, 7% Middle Eastern North Africa. So these are sort of typical results you would get. You'd get some percentages on basically, in a very broad view, where you came from, what your ancestry was. Okay? And so I just want to play a clip here because it goes through sort of two really interesting things, sort of like the thought process behind, well, getting this kind of testing done and how it works a little bit, and what you might want to use that information for.
A cottage industry of genetic testing companies has been sprouting up on the internet, capitalizing on the data generated by the Human Genome Project. At a cost of about $250, you can satisfy a basic human hunger and learn more about your genetic origins. New York Times reporter Amy Harmon has been writing about recent genealogical developments.
Basically, humans migrated out of Africa about 50,000 years ago and settled on different continents around the world. And enough differences evolved in the populations on those different continents that scientists have now found a way to analyze a cheek swab of your DNA and figure out roughly in the world where your ancestors came from.
Some scientists and scholars are quick to warn that the genetic database is not yet complete enough to warrant accurate results.
Africa is a population of over 700 million people. And for geneticists to do some sampling of that population, they go broke. So what they do is take some small subsample of some small subsample, and then make the claim that these kinds of samples reflect something called African ancestry.
It's fascinating in terms of how people begin to identify themselves, but it gets particularly interesting because as a society, we're used to defining race basically by how people look. And we have these mechanisms like affirmative action that are really meant to address things like the need for diversity or past discrimination. Yet now you have people who might look white, but their genes are telling them that they are some percentage of Native American or African American, and do they deserve to qualify for these things?
Who's entitled to regard themselves as Jewish or any of a number of groups? Well, the argument's going to come, partly, it's in the DNA. And others are going to say, no. Being Jewish or being non-Jewish isn't just a matter of whether your DNA tells us you're Jewish or not. It's about whether or not it's the belief systems, the religion. So I don't think the DNA is as definitive when it comes to cultural membership or group membership as people think it is.
Any comments, questions? Do you think that's a good use of ancestry information, of sort of checking the right box on your application? Why are you laughing? Because you don't think it is. I mean, I'm asking really, what would be a good use of that ancestry information? How would you use it? Let's say I gave you a free test, and you could do this. How would you use that information? What would be most interesting to you? So basically trying to confirm or see whether where you think you come from is actually where you come from, that kind of thing? How many people sort of share that kind of... How many people would want to have that information about themselves? How many people would want to pay $250 for it? Okay, yeah. So it's going to get cheaper, clearly. But yeah, I think as is the case with a lot of technologies like this, people sort of rush into it without thinking like what they'd actually use it for. So if you're thinking about doing it, think about also like what information it could actually give you, what you would want to know, and whether that information is accurate, right? So one of the points made in that video clip is that there are some problems with trying to infer ancestry from genes. First of all, it's not entirely accurate. It doesn't have a huge resolution. It can't say your ancestors came from, London. It can't say your ancestors came from, Amman, Jordan. It can say that the probability is high you had some amount of ancestry from Central Asia, from West Africa, and so on. But as we'll see, that the way these tests are currently done, that's only very, very partial information. So I want to go through how the information is actually processed, how you actually get information about ancestry from your DNA. But I want you to be thinking about sort of, whether that's good, whether that level of information is actually useful to anyone. Okay, so I think I mentioned this in a previous lecture that the two most common sources of DNA that are used for ancestry tracking are the Y chromosome and the mitochondria. And the reason is that the Y chromosome is inherited entirely through males, okay, and the mitochondria is inherited entirely from your mother. So males and females both have mitochondria, but you only get your mitochondria from your mother. So basically the mitochondria is a readout of your maternal lineage, and mitochondria have their own DNA, right? So it's a readout of your maternal genetic lineage, and if you're a male, the Y chromosome is a readout of your paternal lineage, okay? And so basically what happens when you do one of these tests is you take your DNA on a cheek swab, you send it to the company, or as you'll see, the National Geographic, and they isolate your DNA, and they take particular sequences from your mitochondria or from your Y chromosome, and they compare those to a database of other sequences from the Y chromosome and mitochondria to sort of place where you fit. Okay.
What if you were interested in a woman's maternal lineage since she doesn't have the Y?
Can't get it. The way you would have to get it would be from her father or from her brother. Right? So this is the way that this information is translated into an actual ancestry. The idea is that, as you'll see in a little bit, the history of our species now, at least in broad strokes, is quite clear. Our species originated in Africa, so in a very real sense, we all have African ancestry. Okay, and so if you think about what's shown here is, for example, the Y chromosome. We can all human males today can trace their Y chromosome back to a single individual male who lived in Africa sometime around a little more than 50,000 years ago. Okay, so if we start with that Y chromosome, that's sort of the original Y chromosome of our species, at least in terms of everyone who's still living today. At certain points in time, mutations happen on that chromosome, and they happen in people in particular parts of the world. Okay? So as you'll see, there was a large migration out of Africa that basically populated the rest of the globe. Okay? And so if along that migration a new mutation arose, that would be a way of distinguishing someone who has ancestry that can be traced to that particular part of the migration. Okay? So around 50,000 years ago, you have a mutation called M168 that arose, and it basically only occurs in people who can trace their ancestry to that migration out of Africa. Sometime later, around 40,000 years ago, you have another mutation called the M9 mutation, okay, and that arose in a single man, right, in the Middle East or Central Asia, right, and so you can tell something about a male's ancestry whether he has the M9 mutation and the M168 mutation or only the M168 mutation. Okay? If it's only the M168, then you can trace his ancestry to this out of Africa migration, but you can't trace it to the Middle East or Central Asia, whereas if he has both, you can trace it to the Middle East or Central Asia. There's another mutation called M3, which occurred in a male sometime around 10,000 years ago. And that mutation is rare in humans because it basically occurred in a male in the population who crossed the land bridge from Asia into the Americas. Okay, so the peopling of the New World was basically through a migration that occurred from Siberia to Alaska when those were basically connected. And so if someone has that M3 mutation, then they have a very high probability of basically Native American ancestry. Okay? And if you collect enough of these mutations, and you can do the same thing on the mitochondrial DNA, then you can start to draw maps of migration routes, because you can say, you know, people who are sort of trace their geographical origin to a particular part of the world tend to share particular mutations, and those are related to other people who might differ only in one of those mutations. Okay, and by tracking these mutations as they arise, as our species spread out of Africa, you can basically draw a map. Okay, and you get a map that looks something like this, and I'll show you this again on another view. But what's depicted here is, well, let's say we want to sort of compare two people, a European man and a Native American man, right? The way we do it is we do this test, right? That we send their DNA off, we get their Y chromosome genetic profile, and you find that the European man has this mutation, this mutation, this mutation, this mutation, this mutation, this mutation, this mutation. Whereas the Native American has these mutations, but then has this one and this one. Okay? And just by that information, you can get a broad view of the ancestry of those two men and where it diverged. Okay? So because these two men share this mutation, this mutation, this mutation, and this mutation, then their ancestry tracks to this migration out of Africa into Central Asia, and it only diverges at this point in time, 35,000 years ago in Central Asia, because that's when the mutations start to differ between them, right? And these mutations are associated with this migration across Asia into Alaska and down into North America. And these mutations are associated with migration in the opposite direction, out of Central Asia into Europe. Does everyone understand that basic concept of how we can use these mutations to sort of track the history? Okay. So I'm not going to play any of these clips, but there are a bunch of clips on your DVD that go along with these ideas of tracking human history and what the DNA evidence tells us. And so what I want to introduce to you now is this, this big project run by the National Geographic Society called the Genographic Project. Okay? And so they are one of these places where you can send away your DNA and your money, right? And then they'll tell you something about your ancestry. And in particular, they use the Y chromosome and the mitochondrial DNA. So if you're a man, you get both tests done. If you're a woman, you only get the mitochondrial DNA done, obviously. Okay? And so they have a really good website. And so if you go to the website and you click on Atlas of the Human Journey, you can basically get a series of these maps, and they're sort of progressing in time. So back to the origin of our species about 200,000 years ago, to the migration out of Africa and afterwards. Okay? And you can get these snapshots of these migrations. Okay? And so if you click through these different time points, you can see, for example, I don't know how well you can see, but what you have are basically migration routes in Africa. Because this is around the time of the migration out of Africa, you see the lineage coming out of Africa, going into basically southwestern Asia, and then along southern Asia to Australia. Okay, so it's about more like 40,000 years ago that our modern human species reached Australia and was separated there for quite a long time. And so if you click further, then the map gets more complicated. Right? So now you see, you know, just 5,000 years later, basically, you see the map expanding. Okay? And if you go all the way towards the present, you can see sort of the more complex migrations that happen, because they're not just like sort of a spreading, but there's migrations and then remigrations and migrations back, and it all starts to get really complicated. I really encourage you to go through this website. So in addition to the maps, they have these videos that you can play along that sort of animate places on the map and point to things. So I just want to play a little bit of this one.
The tree of human genetic diversity has at its root Adam, the common male ancestor of every living man. Because he lived in Africa some 60,000 years ago, all humans must have lived there until at least that time. Unlike his biblical namesake, this Adam was not the only man alive in his era. Rather, he is unique because his descendants are the only ones to survive to the present day. Africa's San people, the hunter-gatherers, once known as Bushmen, carry more ancient evolutionary lineages in their DNA than any other people and exhibit a direct living link to Adam. Their language, Shu, is another link to our ancient ancestors. Shu's speech is characterized by clicking sounds used for word construction, which sounds like those used to guide a horse or simulate dripping water. Shu employs an amazing 141 distinct sounds, while 2/3 of the world's languages, including English, use only 20 to 40. Such linguistic complexity suggests the ancient origins of the language and its sound users in much the same way that genetic lineages become increasingly diverse with age. The archaeological record shows that humans lived in Australia 40,000 or perhaps 60,000 years ago, a very short time after they left Africa. Their monumental journey was made possible by the shifting land masses and lower sea levels of the glacial Pleistocene.
All right, so I'm just going to cut that off because I really encourage you to go to the site and sort of play through the whole history. It's basically the history of our species as we currently understand it. One of the really interesting, I think, valuable things here is that what they're doing, at least in their narration, is integrating the genetic data with anthropological data, with archaeological data, with linguistic data, right? And I think it's really the combination of all those things that's eventually going to give a very rich history of where our species came from and where it went. Okay, so I want to play another clip from the New York Times, which again describes this project and interviews some of the people who are involved in it and the kind of information that you get out of it.
This is Amy Harmon of the New York Times. It is a question that has long occupied the thoughts of scientists, theologians, philosophers, and at some point probably just about all of us.
Where do I come from?
Dr. Spencer Wells is a Harvard and Stanford-educated population geneticist who says that the answer may lie closer than we think. The answer, he says, may lie
inside of ourselves, inside of our bodies, in our DNA.
Working with the National Geographic Society and other scientists around the globe, Dr. Wells is hoping to decipher the hidden history within our genes.
Our genetic code reveals the details of how our ancestors got from an African homeland, 50 to 60,000 years ago perhaps, to wherever we might live today.
The 5-year effort, called the Genographic Project, began last year collecting DNA samples from indigenous groups whose isolation in far-off places like the Tibesti Mountains of Chad and the farmlands of Tajikistan makes them appealing subjects for the study. They are also working closer to home. In July, I joined up with Dr. Wells' colleague and the project's North American director, Tad Schur, on a visit to southwestern Alaska. There, on the bank of the upper Kuskokwim River, Dr. Schur and other genographic staffers met with members of the tribal village of Georgetown and took their cheek swabs to gather the raw data that they hope will tell us more about where their ancestors came from and how they ended up in this part of the world.
In terms of populations he's actually sampled, he sampled the Seekonk Wampanoag, a group living in Massachusetts and Rhode Island, and then several populations living in Alaska.
Since most of the world's 6 billion people are of mixed ancestry, working with these indigenous groups, Dr. Wells says, is key to the project's success and forms the basis of its urgency.
We have to work with these indigenous traditional peoples while they still have a sense of who they are, while they can still tell their side of the story and contextualize the genetic results. So, we do have this closing window of opportunity because when we lose that sense of cultural and geographic context, that sense of connection, the cultural connection with the previous generations, it's very difficult for us to interpret the genetic data.
The ultimate goal is to collect 100,000 DNA samples from indigenous people, as well as to provide all of us with the opportunity to learn more about where our ancestors came from. Although it is not likely to generate a lot of scientific payoff, the project is offering a home DNA test kit that can be purchased for $100 from its website. About 175,000 kits have been sold so far. After sending in their cheek swab, participants can log on to the site and get a glimpse of how and where their ancestors lived beyond the mists of recorded time. More details, the project promises, will be filled in as its scientists learn more from the indigenous samples they collect.
This is a chance for everybody around the world to participate in this real-time research project, which doesn't happen very often.
And what exactly would you learn from sending in your own DNA sample?
It will tell you something about the genetic composition of your haplotype, which is your particular set of genetic markers, and then tell you how that particular haplotype fits into the broader pattern, this tree of humanity, and how your ancestors, again, migrated from an African homeland to wherever they lived most recently.
Undertaking a project like this would have been a lot more difficult just a decade ago, before the Human Genome Project provided scientists like Dr. Wells with a toolkit for understanding our genetic heritage.
The incredible technology that's come out of that has allowed us to start to use DNA as a tool to track historical migrations, and we do this by picking out little positions in the genome that differ from person to person. And these differences allow us to trace back to common ancestors, so they allow us to define lineages that link up people from all over the world.
Finding those links is made possible by the creation of genetic markers that randomly occur over time in our DNA, and provide a sort of timestamp to allow scientists to look back into our genetic history.
So we study the patterns of these genetic markers that link people up into lineages. If you share a genetic marker with someone, you share an ancestor at some point in the past, the person in whom that change first occurred.
Naturally, Dr. Wells has submitted his own sample to the database.
What I found is, perhaps not surprisingly, my father's side of the family comes from England. I have a very typically English Y chromosome. It's a lineage we call R1b, and about 70% of English men are carrying this Y chromosome lineage, and it traces back, obviously, to England, but before that it was in Spain. Before they were in Spain, where were they living? Central Asia, of all places. So this was an early expansion into Europe, possibly the very earliest expansion around 35,000 years ago that came not directly out of the Middle East, but via Central Asia.
All right, so what I want to do now is actually take you through the whirlwind tour of our species history. Okay, and it starts about 200,000 years ago in Africa when the first modern humans are evident in the fossil record. And this migration out of Africa occurred somewhere around 50,000 years ago. And as I said before, basically, in any case where you have a migration like this, where a small percentage of the population is moving somewhere else, you have what we usually refer to as a bottleneck. That is that not all of the genetic diversity that existed makes it into the migration into the rest of the world. Okay, and that's true of any species you want to think about in terms of migration. So what's shown in these pictures, which are from this article that you'll read, is the different colors are meant to represent different sort of genetic variations that might exist in the population in Africa. And what happened in the migration out of Africa is some small proportion of that variation was carried by people out of Africa. So when we say that we're all descendants from Africa, basically the strongest evidence for that, in addition to the fossil record, is that the greatest amount of genetic diversity of our species is in Africa. Okay? And that'll be true whenever you have a species that sort of expands out of somewhere. The original source location of that species is where you'll find the largest amount of genetic diversity. So if you follow a little bit longer, it didn't take that long in the scheme of things. So another 10 to 20,000 years for the human population to be basically worldwide. And as you can see, again, it's this limited amount of genetic diversity that exists in Africa that's now existing all over the world. Okay? We know something now about the various migration routes. Okay? So this is a figure from that article, and we'll just zoom in on a few places. Okay? So again, origin of modern humans about 200,000 years ago in East Africa. The oldest modern human fossil is about that age and from that location. Now, when humans migrated out of Africa, there were other human-like species in existence at the time. The one you're probably most familiar with is Neanderthals. Okay? This is an artist's rendering, I think, or unfortunate actor who's seen better days, of what a Neanderthal might have looked like. Okay, so when humans, that is our ancestors, modern humans, migrated out of Africa, there were other humans like Neanderthals around. And so what happened after was pretty clear. Neanderthals no longer exist and we do. Okay, so at some point the Neanderthals were wiped out probably by modern humans. The peopling of Europe is about 30 to 40,000 years ago, and previously, before genetic evidence, there was some doubt about where that happened. It was originally thought, for example, that the migration to Europe actually was directly from North Africa, okay? Because as you know, that particular point, the Strait of Gibraltar, is not very wide, and so it could have been that the peopling of Europe was actually this way. But the genetic evidence is very clear that the migration out of Africa went into Asia and then went to Europe. As I said, Australia got settled very early after the migration out of Africa. So somewhere 40, 50,000 years ago, the first modern humans arrived in Australia. The peopling of Asia happened through, again, Central Asia, the steppes, north of the Himalayas, and then southeast to China and Japan, east to Siberia, and then again across Siberia into Alaska. At that point in time, there was a land bridge connecting the two, so it was literally basically walking across about 15 to 20,000 years ago, and that's the settling of North America. What you'll read in the article, and so what's interesting in this field is how quickly things move, right? So what you'll read in this article that was written in 2006 is they'll have a list of sort of unanswered questions. And some of these are unanswered, but some of these are actually answered now. All right, so this is a list of questions that they pose. Which African populations are the most ancient? What was the impact of migrations into Africa, so back into Africa after humans left Africa? Did particular historical events, like the conquests of Alexander the Great or the Roman Empire, leave a genetic trail? Is there genetic evidence of Central Asian migration into India? So how did India get populated? Was it up into Central Asia and then back down, or was it along the southern coast? When did people first reach Japan and Taiwan? So on, so on, so on, so on. When did people first reach North America? When did people first reach South America? Can DNA from ancient bones be studied? So if we find a fossil somewhere, can we actually get the DNA from that fossil and learn more than just the indirect evidence of people who are alive today? And what will that tell us about human migrations? So DNA from ancient things is now commonplace. Okay, so here's a timeline from, this is just to give you an example of how some of these questions are now answered. So this is a timeline of using ancient DNA to answer questions about various extinct or existing organisms. So Neanderthals were first discovered in the 1800s, and so the classic Neanderthal specimen was from 1850. It wasn't until 1997 that the first genetic data from Neanderthals came around, and that was sequencing of the mitochondrial DNA from an actual Neanderthal fossil. People were worried at that time that would be the best we could do, because nuclear DNA, that is your normal chromosomes, were found to be always degraded in samples like this. It was hard to get any amount of DNA out of them, and so it was thought that maybe mitochondria would be all we could get. But then a cave bear and a mammoth were actually sequenced. The normal chromosomal nuclear genes were sequenced with new technology, giving some hope that maybe we could go back to Neanderthals and actually get some more genetic data from them. So in 2006, the Neanderthal Genome Project was established. Okay, only 6 years after the Human Genome Project was completed, we're on to Neanderthals. In 2007, a lot of progress had already been made. 1 million nucleotides from the Neanderthal genome had been determined, but then it was found that a lot of those were actually contaminated, so maybe not so much as 1 million. This is difficult business, right? You find a fossil, the DNA is really degraded. It's hard to, a lot of people have touched those bones. It's hard to separate out Neanderthal DNA from human DNA. The first full genome from an ancient species, so the mammoth, which no longer exists, was published in 2008. Different genes were found to be present in Neanderthals that indicated that they had red hair and fair skin. Okay, just by looking at which alleles they have of particular genes, you could tell what they look like. It was found they also have a version of this gene, FOXP2, which is a transcription factor, that was the same as us, and this gene has been implicated in our ability to have speech. Okay, so this is evidence, maybe the best evidence we'll ever get, that Neanderthals could actually speak to one another. Complete Neanderthal genome finished 2009.